time of the 2003 at the Human Genome Project finished to sequence all Homo sapiens genome, we were like a bit disappointed about one thing. We do have the same amount of genes than onions. We have 2 times the amount of gene of a fly. We were disappointed on our anthropocentric view of ourselves. But there was something we were ignoring, is that we carry on us and in us 100 trillion microbial cells. We are not human. You are a walking microbial ecosystem. That's why we were a bit disappointed about the number of genes, but now it's feeling better. So all this microbial ecosystem is called the microbiota. And all this microbiota, if you're taking into account the 2 million genes, it's called the microbiome. It's all the genes we are encoding in the microbial cells. And this is huge. This is 25 times the human genome size. And here we sequence not the exome, it doesn't exist. We do not target anything. We all shotgun sequencing it all. So Jen sometimes complain about big data. We complain more about it. So the human microbiota, there are different species for different reasons. So first and foremost, I would like to say that microbial ecosystem on you, in and on you, are very specific, right? But it's very stable through life too. It's stable. It can change through the burning process. It can change in adolescence. It can change through your life if your woman are pregnant. It can change when you get age. But it's stable. It's resilience. It's huge resilience. It can be disturbed, but as soon as disturbed a bit, it can be reached the equilibrium again. And this microbiota co-evolve and coexist with every of us for million years. That's also very important to say it. There are bacteria in, on, or on us that are specifically to us. We do not find them anywhere else. Few examples about it, like oral microbiota. It's about 700 species. It's not too much, 700 species, bacterial species. Did you know that like partners tends to have a more similar mixed microbial ecosystem in the oral than non-relative people? And this has been shown to be linked with the kiss frequency. So 10-second kiss, you exchange 80 million bacteria. That's huge. Some stuff about the skin. The skin makes you more attracted by the mosquitoes. The mosquitoes tend to bite more people because of their microbial ecosystem on their skin, because of the metabolites they produced. Some about the original tracts, the vagina. Pregnant woman develop a specific microbial ecosystem in the vagina from the first trimesters of the pregnancy. And what it changed, there are one kind of bacteria called lactobacillus, which we found only on the gut, who starts to develop on the vagina. And why that is to prepare the baby to digest breast milk. I find it very, very beautiful. Then our ecosystem, the microbial ecosystem that is developing on the gut, that's for far the most complex ecosystem in our body side. It is about 300 species for all of us, but it's about like 10 to the power 12 cells per gram of contents in the intestine. In the stomach, it's more or less, it's 10 to the power 1, but it's a huge complexity. And for a very long time, we are not able to study this because of this complexity of this ecosystem. But NGS came as a golden age for gut microbiome research. Just for you to just bring some historical data set here. Microbial cells been first observed 500 years ago, not so far from here in Delft, and with the basic microbial tools, microcosm. And then 200 years ago, the first bacterial cells been isolated on agar plates. we were happy. We're starting to study the bacteria because we were able to observe them. We were able to isolate them. But one thing we realized 100 years later, just after Fred Sanger came with his first sequencing technology, and it's have been expressed by Stanley and Knopka, about the Great Plate count anomaly. What is this Great Plate count anomaly? We find out that we observe much more bacteria under the microscope than in the agar plate. Was tremendous. We were we were studying with the agar plates, the Petri dishes, only 30% of the community present in the guts. And this was true for all the environmental sample. Most of the bacteria we cannot isolate. On the guts, there are two reasons for that. It's anaerobic. It's very hard to find anaerobic condition and cultivate and isolate the bacteria, but also because bacteria depend to each other too much to grow in isolate form. There are exchange of metabolites in between bacteria a lot in every environment. And it's very hard to understand which metabolites the other bacteria produce than the one needs to use to grow. But on that time, the next generation sequencing revolution come, and with that, our ability to sequence the full genome of the bacterial presence in the guts. And the first study was published in 2006. There were two studies in the lab of Jeffrey Gordon about the first result we get is Jeffrey Gordon expressed us as a super organism, telling that he was finding so much of the diversity there. I was amazed about that. And the second one was done on the mouse guts, and they linked the microbial ecosystem to obesity. And since this time, and with the decreased price of costs per raw megabase of DNA from the next generation sequencing development, a lot of international projects specific on the guts, such as MetaGenoPolis in Europe, but also American guts in USA, and more largely the human microbiome project starts to grow and to having more and more cohorts and more people involving in cohort study to sequence the bacteria which were present on their guts, but also sequence the bacteria who were present in every body habitat and site. So us and our guts, what we do for them, and then we will see what they do for us. So from the birth times, depending on the way we burst, it will change microbial ecosystem in your gut. Depending if you're born by cesarean or vaginal way, this will impact, at the time you're born, it's already impacting a lot your gut microbial community. And then the first milk you will have, is it a formula feed milk or is it a breast milk? This will also impact a lot on the gut microbiota you develop. The gut microbiota developed the first 3 years of age. And then is stable. So this is very important, and this is very deciding for the mixed microbial community we will have in the guts in the futures. The diets also, of course, the number of carbohydrates, the number of fiber you have in on your diets, it's very important. Then the environment, if you are sportive or not, if you smoke or not, do you have stress life or not? This will also impact gut bacterial community in the guts. The disease states also impacted. Aging, it have a huge impact. Huge impact from birth to adolescence and then to old people. You don't have the same mixed microbial community through age, even if it's stable. But some species appears and some species time to be more abundant at that time. Drugs also imply a lot on your gut, bacterial community. The geographical area where you come from, we call it the westernizations. It's linked to the diets, of course, but infants in the rural village in Africa have much more richness than European infants in the guts, in terms of species, but also in terms of genes. Now, what the bacteria do for you? They do a lot. They help you to digest the food, of course, but they also biosynthesize specific amino acid and specific vitamin that your body are not able to synthesize. They play a big role on the drugs. That's very important. Do you know that, we heard about Andy speaking about the immunotherapy, how this immunotherapy impacts the guts. And we just worked together with research centers on the cancer next to Paris, the Gustave Roussy Institute. And we work on them, and we observe the gut bacterial community change through immunotherapy. And what we find out is that the immunotherapy efficiency was based on the bacteria you have on the guts. So Andy said to us, yeah, it's depending on the immune system you have. And and say, yes, indeed, and it's depending on the bacteria you have in the gut. Why? Because the bacteria develop in parallel with the immune system together, and they define how you are protected against the disease, against the pathogen. And this will be the second part of my talk. I would like to emphasize, your bacteria is good for you. They protect against pathogen. Why? It's because the bacteria and the immune system develop together through the first years of age. So here we go. So healthy microbial communities that not very well defined. We don't have normal microbial community in the gut, because we are all different, but healthy one is not defined yet. But for sure, the commensal microbiota, we call it the commensal microbiota because we know they are not, they do not cause disease or pathogen and so on, fight against the pathogen. They fight against the pathogen in a direct way. For example, some bacteria, such as Bacteroides here, will consume the monosaccharides that the pathogen Citrobacter will use. So consuming this monosaccharides, it will have a direct impact on the colonization to the Citrobacteria's pathogen bacterial. But then you have a whole bunch of immune system driving resistance against the pathogens. And this is a bit more complex, but look, this similar Bacteroides just will make some impacts on the abundance of the reg3 proteins productions, will directly impact the growth of Enterococcus antibiotic resistance. And how it happens, it will impact on the immune system, such as the toll-like receptors, production of interleukin, which will also play a major role in the production of reg3 gamma, which will fight against the Enterococcus. So this is very important to understand. You have the host immunity resistance, mediated resistance. So then, about disease. At the point of this talk, you should ask, why? These guys talk us about what protects us from disease and not what affects us. But that's a very important point, and this is what you have to understand, is that from the beginning, our gut bacteria is good to protect us. But indeed, you have disease a lot linked to the gut bacterial community, and I will come to this point now. So since these 60 years, medical progress did a lot to decrease a lot the incidence of hepatitis A or tuberculosis. It was great. I mean, it's last years of medical progress. But in the other hands, the immune disorders, relative disease, just keep increasing a lot. Like take the type 1 diabetes, take the multiple sclerosis, take the Crohn's disease, the inflammatory bowel disease. All these diseases increase a lot these last 60 years. We count that 1 in 4 people will be or are infected by immune disorders right now. We don't know exactly how to cure them, but what we know for sure is that it's linked to the host's bacterial links in the gut, but also maybe through different body sites. And if we want to list and to classify the disease that we know are impacting, you will divide them through diet and the microbiota links, or diet and the immune system links, or in between microbiota and the innate immune system. And we will classify in 3 groups. First, the metabolic disorders, such as obesity, arthritis, or non-alcoholic fatty liver disease. This is all about metabolic disorders and infecting such as the liver and the arteries. But also you have the cancerogen disorders, which are neoplastic disorders, which is the growth of a tumor, and which is a particle disorders linked to the triptych between diet, microbiota in the gut, and innate immune system. And then you have a group on autoimmune and inflammatory disease, which will impact the gut, of course, with the inflammatory bowel disease. It will impact also the pancreas with the type 1 diabetes, the skeleton with the arthritis, and the pulmonary disease, and so on. And still, these figures miss the brain-gut axis. If you take into consideration the brain-gut axis, then you can add autism, Parkinson, Alzheimer's disease, all been having association with the gut bacterial species and the genes which are inside our guts and carry on by these bacterial species. This is huge. I just list 2021, but there are more coming every year. And it's all about gut dysbiosis. What is a gut dysbiosis? The gut dysbiosis is at one time the equilibrium and the homeostasis in between the pathobionts and the commensal bacteria, the symbiont, is going to be disturbed. It will be different factors, and I will go to tell you more about it. And so you have the pathogens. The pathogen is one way to see it. Yeah, the pathogen is there, then there are disease. But I would like you to change a bit your way of thinking. It's not about presence of pathogen or not, because even in the healthy case, the pathogen is also present, but the commensal bacteria fight against the pathogen, and they are doing resistance. And as I am being going as a microbial ecologist on environmental science, I would like to give you an example and to tell you what it is, the dysbiosis, to compare with eutrophication. Eutrophication. You have a lake, you have fish, you have oxygen, you have waters, you have sun, you have everything in homeostasis, as you have homeostasis in the gut at the time of the equilibrium. But suddenly, you add some pollutants, you have some phosphates, you have some azote coming from the farmer, and suddenly, they have a bloom on the algae, everything grow, you have lower oxygen amounts in the lake, and all the fish died. And you just disturb the system completely. It changed from one way to another. You don't have any life there, only plants, but not anymore fish, a lot of bacteria too. So the system is completely unbalanced, and it's because of one disequilibrium. For dysbiosis in guts, it's exactly the same. You have environmental factor and genetic factors. We are not sure yet which one play a role. I think it's about the both play the role, and the both are important, but they are one perturbation at one time in the life who happen and cause dysbiosis. And dysbiosis cause, for example, decrease of commensal bacteria, which were there, and to fight against the pathogen. And then the pathogen find about a niche, an ecological niche to develop. And then they will fragilize your epithelial cells, and then we call it the leaky epithelial cells, because the epithelial cells will not be tight in between each other, and then the pathogen will cause disease. And this is, as you can see on the panel on your right, this is linked to a lot of regulation of immune system, of course. So a bit more about the Institute, MetaGenoPolis in INRA, and the way we're going to do analysis, for sample collection to data analysis. So the sample collection, we receive stool at the lab, and stool collects following our advice and consign, because it's very important. All protocols are designed on the international human microbiome standards. If we want to compare the data we have to each other in between different labs, it's very important that we standardize the method. DNA extraction only have found to impact a lot on the diversity you can find on the guts. So for that, our lab, in collaboration with a lot of other labs, published in Nature Biotechnology this year DNA extractions that have to be applied for every gut sample if we want to be able to compare the data we receive. So as soon as the stool sample arrive in the lab, we do DNA extraction, show full DNA extraction, and then shotgun sequencing, no targets, no amplification of specific taxonomical markers. We do shotgun sequencing. We have 4 different protons. We have a huge, of course, high throughput. We have 4 sequencers. We do 8 runs a day. We sequence about 640 million reads a day. It's about 30 times the human genome. So, of course, we have a need that other platform doing human genetics do not have. For one sample, we try to cover it with 20 million. We do cover it 20 million reads. That's the deepness of sequencing we need to cover a single genome present in the microbial ecosystem in the guts. And we treat about, right now, 3,000 samples a year. It's about 12,000 samples we've been treated since 2012, and we have the capacity for 6,000 samples a day. And then we use a reference catalog, exactly at the same than geneticians use the human reference genome. We use our genome reference catalog. This catalog has been built using 500 different individuals' gut bacterial community and been enriched years after years. We get now 10 million genes in these catalogs, and it's 10 million genes are found in an individual. From these 10 million genes, only a few fraction of it are found to be common in between people. So we map directly the read we sequence to these reference catalogs, and we end up with a table which counts the genes inside the DNA we've been sequenced. And we have a table of gene counts. And then these have been, of course, analyzed with a bunch of lot of bioinformaticians and analysts and data analysis. And everything is well automatized, of course, because we treat a lot of samples through the year. We have only 2 people in the sequencing lab. So a few things about what we've done since the MetaGenoPolis project. We've been born from it. We publish so first student collaboration with EMBL in Germany, where we published the first catalog in 2010. It's been covered by Nature as our other genomes, which were much complex to study, and since we're still going to be surprised about it. So we are surprised. We developed the method, so it's first in 5 years ago it was 3.3 million genes. Now it's 10 million genes. Keep enrich these catalogs a lot, going with more and more individuals. We also developed methodology to reconstruct the genome of bacteria. What are we using for that? We use the co-abundance of different genes in between people. And using this co-abundance of genes in between samples, we reconstruct the genome of the bacterial or the biological units, we will call it, because it's not always the bacteria, but and that's the way we do. Then we have a way to stratify, because the data is so big, so complex, we need to stratify them. And to stratify them, we tried first on the first study published in 2011, enterotype of the human gut community. So it tells that every one of you have a specific microbial ecosystem and can be divided in 3 different groups following the bacteria abundances. And then a better way, I think, to stratify people, we find out that some people have very rich genes and some people are low genes richness. So, and this also stratify people, and it can be used, a way to use it to do diagnostic. And then we work on a lot of different diseases, such as type 2 diabetes, which we find out 60,000 different biomarkers, which were different in between control and type 2 diabetes. We work on the liver cirrhosis. Last year, we worked on the control, on the insulin resistance linked to type 2 diabetes. We found out that one bacteria was more abundant in the insulin resistance compared to control. And why is that? It's because in this gut bacteria, so insulin resistance, you have a lot of branched chain amino acid accumulation in the blood of these insulin-resistant people. Why? It's because you miss one bacteria to transport this branched chain amino acid, but you have these bacteria who produce them. That's why accumulation of branched chain fatty acid linked to the insulin resistance. And that's why it's very interesting, and I go with my last slide now. So we have found a lot of correlation. We are not yet there to say causations. And this is what we want to go, and that's our visions. We want to do longitudinal study. We want to have much more medical data collection and links the data we found on the gut with the serum, with the urine, with the saliva, the gene, the metabolite present there. We also want to integrate metagenomics and metabolomics. So the genomics is the potential, the metabolites is the output. We want to link these two more and more. And also using maybe a technology that I developed during my PhD thesis, which is involving to take a sample and to extract sequentially DNA, proteins, RNA, metabolites, extracellular and intracellular from the same sample. Why? It's because we have a lot of heterogeneity there, and we want to avoid the bias in between integrated multi-omic data sets, so we start from a sample the smallest as possible. And for that, we need to involve clinicians and medical doctors, partners, more and more. And with that, I would like to thank all of you in the room, but also in the live streaming, and take good care of your gut microbiome. Thanks.